The multi-photon fluorescence recovery after photobleaching procedure is easily demonstrated with a freely diffusing sample of fluorescent dye molecules. First, a low-intensity laser beam is sent into the sample to generate a reference level of fluorescence from the dye molecules within the focal volume of the laser. Next, a brief high-intensity laser flash is sent in, photobleaching a portion of the dye molecules within the focal volume. Finally, the laser beam is attenuated to the previous low-intensity level, and the fluorescence is monitored as photobleached molecules diffuse outward and are replaced by still fluorescent molecules from outside the focal volume. The resulting fluorescence as a function of time curve is analyzed to yield the diffusion coefficient.
Hi, I'm Kelley Sullivan from the laboratory of Edward Brown in the Department of Biomedical Engineering at the University of Rochester. Today, we'll be showing you how to perform multi-photon fluorescence recovery after photobleaching. We use this technique in our laboratory to study diffusion in both in vitro and in vivo biological systems. So, let's get started.
To begin, set the desired laser wavelength and insert the appropriate filter in front of the photomultiplier tube, or PMT. Next, to avoid damage to the Pockels cell, adjust the laser power to 200 milliwatts or less. There are two mirrors, oriented in a figure 4, upstream of the Pockels cell. These mirrors are necessary and sufficient to align the laser beam through the Pockels cell. Set the voltage offset on the Pockels cell bias controller to 0. Adjust the laser path through the Pockels cell such that the laser is centered in the front and back windows. Use the mirror upstream in the figure 4 to center the beam in the front window, and the mirror downstream in the figure 4 to center the beam in the back window. Now set the voltage across the Pockels cell crystal to the maximum value. The appropriate value is wavelength dependent and can be found in the Pockels cell instruction manual. Adjust the mirror